Welcome to the Deep Dive, your shortcut to being well-informed. We cut through the noise and get straight to the core of fascinating topics. Today we're digging into something fundamental but maybe overlooked, how plants actually move stuff around inside them. Water, nutrients, you name it. We're pulling the key insights from a chapter in the classic Raven Biology of Plants, and our goal is simple. Give you a clear grasp of these amazing systems, no textbook needed. I mean, think about it. Plants seem so still, right? But inside, they're like these incredible hydraulic engineers. They're moving huge amounts of water and nutrients, sometimes over enormous distances. Imagine a giant redwood pulling water hundreds of feet up. We'll be focusing on the two main transport tissues, xylem and phloem. And their jobs are actually more connected and surprising than you might think.
Yeah, it's fascinating how long it took us to really figure these processes out. For centuries, people saw sap moving, but the how was a real puzzle. It wasn't really until, the late 19th, early 20th century that the pieces started fitting together, how water defies gravity, how sugars get distributed. It shows how much we've learned.
Absolutely. Yeah, and some of those early observations are just incredible. Let's get into the water and nutrient movement part first. Picture this. Back in the 1700s, Stephen Hales, an English physician, found a sunflower could lose water vapor, he called it perspiring, 17 times faster than a human over 24 hours.
17 times.
Yeah. And that's just one sunflower. A single large tree can lose hundreds of liters, maybe 200 to 400, in just one day. Yeah. This process, losing water vapor from leaves mostly, is transpiration.
So why? Why lose all that water? It seems wasteful, but it's actually fundamentally linked to photosynthesis. It's sometimes called an unavoidable evil. See, plants need big leaves for sunlight, and they need moist surfaces inside those leaves so CO2 can dissolve and get into the cells. But if you have water exposed to air that isn't saturated, you get evaporation. It's simple physics. So getting CO2 in means water inevitably gets out. Too much loss, though, and the plant is in trouble.
Right. But plants have tricks up their sleeves, yeah? Adaptations to cut down on that loss, like the waxy coating on leaves, the cuticle.
Yeah.
That's a pretty good barrier.
It is, yeah. Very effective.
But the real control points are these tiny pores called stomata. They're microscopic, maybe only 1% of the leaf surface, but they account for, over 90% of the water lost through transpiration.
Exactly. It happens in two steps. Water evaporates from cell surfaces inside the leaf, turning into vapor in the air spaces there. Then that vapor diffuses out into the drier outside air through those stomatal pores. It's all driven by differences in water vapor concentration.
And the way these stomata open and close is just ingenious. Each pore has two guard cells around it.
And it all comes down to changes in the shape of those guard cells, controlled by something called turgor pressure.
Turgor pressure, like water pressure inside the cell.
Pretty much. When stomata need to open, the guard cells actively pump in solutes, mostly potassium ions, K+ in to start, and later, sucrose plays a role too. This makes the inside of the guard cell more concentrated, lowering its water potential makes it thirstier, essentially.
So water flows in.
Exactly. Water rushes in by osmosis, inflating the guard cells. They swell up, increase their internal pressure, their turgor. Because of how they're built, as they inflate, they bow outwards, opening the pore between them. Think of two slightly curved balloons inflating side by side.
Okay, I can picture that. So closing is just the reverse.
Yep. Solutes leave, water follows, the cells lose turgor, become flaccid, and the pore closes up. It's often a daily cycle, K+ in the morning, sucrose later on.
And you mentioned how they're built. The cell walls matter too, right?
Oh, absolutely crucial. The cellulose microfibrils, the tiny reinforcing rods in the cell walls, are arranged radially, kind of like spokes on a wheel, around the guard cell. This means when the cell takes up water, it gets longer and bends outwards, opening the pool rather than just swelling sideways like a regular balloon might. That structure is key.
Clever. And what about the environment? Does that affect this stomata?
Definitely. CO2 concentration is a big one. Higher CO2 inside the leaf usually triggers closure. Plants don't need to keep the gates wide open if CO2 is plentiful. Temperature also has an effect. Generally, between 10 and 25 Celsius, it's not huge, but above 30-35 degrees C, many plants start closing their stomata.
Why is that? Just the heat?
It's partly because higher temperatures increase respiration, which releases CO2 inside the leaf. That buildup signals closure. It's a water-saving move in hot conditions, often leading to midday closure.
And plants have internal clocks too, circadian rhythms.
They do. Many plants will open and close their stomata on a roughly 24-hour cycle, even if you keep them in constant light or darkness. It's built-in programming.
Okay, now this is cool. Siam plants. Cacti, pineapples.
Sorry.
They do things differently, right?
They really do. Crassulacean, acid metabolism, see 'em. It's a fantastic adaptation for deserts. They flip the whole thing. They open their stomata at night when it's cooler and more humid, so they lose less water. They take in CO2 then and store it chemically
Yeah,
as organic acids.
and then during the day.
Stomata are shut tight, but they use the CO2 they stored overnight for photosynthesis, powered by the sunlight. It's a brilliant water-saving strategy.
Amazing. Are there other plants that do weird things at night?
While some regular C3 plants actually don't fully close their stomata at night, this is called nocturnal transpiration. They lose some water, yes, but the thought is it might help them pull in more mineral nutrients from the soil when evaporation isn't as intense.
Interesting trade-off. So, besides stomata, what else affects how fast a plant loses water?
Several things. Temperature. Obviously, evaporation increases with heat, roughly doubling for every 10 degrees C rise, though the evaporation itself does cool the leaf a bit.
Like sweating.
Kind of, yeah.
Yeah.
Humidity is huge, too. If the air outside is already very humid, the gradient driving water vapor out is much smaller, so transpiration slows down.
Explains why rainforest plants can have those huge leaves.
Exactly. They can afford to maximize light capture. Whereas in dry, windy grasslands, you see narrower leaves, thicker cuticles, everything geared towards conserving water.
And wind. Does that matter?
Oh, yes. Wind blows away the little layer of humid air right next to the leaf surface called the boundary layer. This steepens the gradient between the inside of the leaf and the outside air, so transpiration usually increases.
Okay, so plants are losing all this water, which brings us back to that redwood. How on earth does water get from the roots way down there all the way to the top leaves, hundreds of feet up?
That's the million-dollar question, isn't it? We know water moves in the xylem, those bundles of pipe-like cells. Simple experiments, like putting a cut stem in colored water, show the dye moving up through the xylem vessels and tracheids.
But how? Is it pushed from the bottom?
For a long time, people thought maybe root pressure pushed it up, but that's definitely not strong enough for tall trees. The dominant theory now is the cohesion tension theory.
Cohesion tension. Sounds sticky.
It kind of is. The core idea is that water is pulled from the top, not pushed from below. It starts with transpiration evaporation from the cells in the leaves. This loss of water lowers the water potential inside those leaf cells.
Makes them thirstier again.
Exactly. This creates a negative pressure, a tension, or a pull on the water remaining in the leaf xylem. Now, water molecules have this amazing property called cohesion. They stick to each other really strongly, like tiny magnets, because of hydrogen bonds.
Is it a chain?
Precisely. So the tension created in the leaves pulls on this continuous chain or column of water molecules, extending all the way down the xylem through the stem, right down to the roots. And there's also adhesion water molecules sticking to the xylem walls, which helps counteract gravity and keep the column intact. So maybe cohesion-adhesion tension theory is more accurate.
Wow. So the pulling force comes from evaporation at the top.
Yes, and the energy for this entire massive water transport system ultimately comes from the sun, driving that evaporation. The plant just provides the plumbing. There are experiments showing water uptake closely follows transpiration rates, and models using things like porous clay cups attached to mercury columns that demonstrate this physical pulling effect.
That's incredible.
Yeah.
But it sounds risky. What if that chain of water breaks?
That's the weak point, the Achilles heel, you could say. Air bubbles. If the tension becomes too great or if stresses like freezing occur, the water column can break. That's called cavitation. If the conduit then fills with air or water vapor, that's an embolism. And once that happens, that particular xylem vessel or tracheid is basically out of commission. It can't transport water anymore.
So how do plants deal with that? It must happen all the time, especially in dry or cold conditions.
They have several coping mechanisms. First, the conduits are very narrow, which helps maintain the cohesive forces. More importantly, the connections between adjacent conduits have these structures called pit membranes. They're porous, allowing water through, but the pores are tiny. The surface tension of water across these tiny pores is usually strong enough to prevent an air bubble in one embolized conduit from spreading or seeding into its neighbors. It contains the damage.
Like fire doors.
That's a great analogy. Conifers have an even fancier system with a structure called a torus in their pits that can actually act like a plug to seal off a damaged tracheid.
So plants can tolerate some embolisms.
Yes, there's redundancy. But widespread embolism caused by severe drought or freeze-thaw cycles can be fatal.
How do scientists even know this tension exists? You can't exactly stick a pressure gauge into a single xylem cell.
True, they use clever methods. One is the pressure chamber, sometimes called a pressure bomb. You cut off a leafy twig, put it in a sealed chamber with the cut end sticking out, and then pressurize the chamber with gas. When you cut the twig, the water column under tension snaps back into the xylem. The pressure needed in the chamber to force the water back out to the cut surface equals the negative pressure, or tension, the water was under originally.
Ah, okay. Indirect measurement.
Right. They can also measure sap flow velocity directly, maybe using heat pulses. They gently heat the sap in one spot and time how long it takes for the warmer sap to reach a sensor further up. And these measurements confirm the flow starts in the twigs in the morning, following the sun, then moves down the trunk, consistent with a pull from above.
Any other evidence?
Yeah, another cool one is measuring tiny changes in tree trunk diameter. Trunks actually shrink slightly during the day when transpiration and tension are highest because the tension pulls the xylem walls inward. Then they swell slightly at night when tension relaxes. It's minuscule, but measurable.
Wow. So that all points back to that solar-powered, cohesion-driven pull.
It really does. It's a testament to the unique physical properties of water and the elegant structures plants have evolved.
But even this amazing system have limits, right? You mentioned redwoods earlier. Is there a maximum height?
It seems so. Studies on the tallest trees suggest the tension in their upper xylem is getting dangerously close to the point where widespread cavitation would occur. The sheer effort of pulling water that high also creates water stress that likely limits leaf growth and photosynthesis at the very top. The current thinking is the maximum height is probably somewhere around 122 to 130 meters. Gravity and physics set a limit.
Okay, so the water gets pulled up. But how does it get into the roots from the soil in the first place?
Roots are the foundation, literally and figuratively. They anchor the plant, of course, but they also have to absorb enormous amounts of water to replace what's lost through transpiration.
And the key players there are the root hairs.
Absolutely. These aren't separate roots, but tiny finger-like extensions of the root's epidermal cells. They massively increase the surface area for absorption. A single rye plant might have billions of them, creating a huge interface with the soil.
So water soaks in through these hairs. Then where does it go?
It has to cross the root to get to the central vascular cylinder where the xylem is located. It can travel between cells, through the cell walls, that's the apolast pathway. Or it can move through cells, either crossing membranes from cell to cell, the transcellular pathway, or moving from cytoplasm to cytoplasm through little connections called plasmodesmata, the symplastic pathway.
So multiple routes.
Yes, but there's a critical checkpoint. A layer of cells called the endodermis surrounds the vascular cylinder. And the walls of these endodermal cells have a waterproof band embedded in them, the Casparian strip. Think of it like mortar between bricks.
What does that do?
It completely blocks the apoplast pathway, the route through the cell walls. So all water and dissolved minerals must cross a living plasma membrane of an endodermal cell to enter the vascular tissue.
Ah, so the plant gets control. It forces everything through a membrane gatekeeper.
Precisely. It allows the plant to selectively control what ions get into the xylem stream. Some roots might also have a similar layer, the exodermis, further out.
Okay, we established water is mostly pulled, but you mentioned root pressure earlier, a push from the bottom.
Yes, under certain conditions. When transpiration is very low, like at night, and soil moisture is high, roots can actively pump mineral ions into the xylem in the vascular cylinder. This makes the xylem sap more concentrated, lowers its water potential, and water flows in from the surrounding root cells by osmosis, building up a positive pressure from below.
And this causes guttation.
Exactly. Guttation is when you see little droplets of water on the tips or edges of leaves, usually in the morning. It looks like dew, but it's actually xylem sap being forced out through special pores called hydathodes by this positive root pressure.
So it's internal water being pushed out.
Correct. But again, this pressure is relatively weak. It can't push water up a tall tree, and many plants, especially conifers, don't really do it. It's more common in smaller herbaceous plants when conditions are right.
Fascinating. What about this other root thing, hydraulic redistribution? Sounds complex.
It sounds complex, but the idea is simple. Water moving passively through roots from wetter soil areas to drier soil areas. It typically happens at night or when transpiration is low. If a plant has deep roots in moist soil and shallow roots in dry soil, water absorbed by the deep roots can move up into the shallow roots and then actually exit into the drier surface soil. That's hydraulic lift.
So the plant is watering its own surface roots, or even its neighbors.
Potentially, yes. It can also happen downward. hydraulic descent, or sideways, the benefits can be significant. It can keep surface roots and their associated microbes, like beneficial mycorrhizal fungi, hydrated during dry periods. It might share water with neighboring plants, like sugar maples sharing deeper water with shallower rooted plants. And on a large scale, like in the Amazon, it's thought to significantly boost water cycling and influence regional climate.
Wow, roots doing plumbing work outside the plant too. Okay, let's switch slightly to the minerals, the inorganic nutrients. How do they get taken up? Is it just dissolved in the water?
Some comes along with the water flow, but uptake is primarily an active, energy-requiring process. Plants have to pump ions in, often against a steep concentration gradient.
You mean there are way more nutrients inside the root cells than in the soil?
Often, yes. Like pea roots might have 75 times more potassium inside than in the surrounding soil solution. Rutabaga root vacuoles can concentrate potassium 10,000-fold. That doesn't happen passively.
So the plant spends energy on this.
Definitely. There are two main energy-dependent steps. Actively absorbing ions from the soil across the membranes of the outer root cells, especially root hairs, and then actively secreting those ions from adjacent parenchyma cells into the xylem vessels. If you deprive roots of oxygen, stopping respiration and energy production, mineral uptake plummets.
And those fungi you mentioned, mycorrhiza, they help here too.
Hugely important, especially for nutrients that don't move easily in the soil, like phosphorus, zinc, and copper. The fungal threads extend far out from the root, acting like an extension cord, accessing nutrients the root couldn't reach and transporting them back to the plant. It's a critical symbiosis for most plants.
Once the ions are in the xylem, do they just go straight up?
Mostly, yes, carried by the transpiration stream,
Yeah.
but there's also exchange happening between the xylem and the phloem, the other transport system. Ions can move sideways between the two streams.
So nutrients can circulate around.
Yes, to some extent, some ions like potassium, phosphate, and chloride are quite mobile in the phloem. This means they can be easily moved from older leaves, say, to newer growing areas where they're needed more. Others, like calcium, boron, and iron, are relatively immobile in the phloem. Once the xylem delivers them to a leaf, they tend to stay put. This difference is important for understanding deficiency symptoms and even for things like foliar fertilization spurring nutrients on leaves.
Okay, that brings us perfectly to the phloem.
Hmm.
We've done water and minerals up the xylem. What about the food, the sugars? That's the phloem's job, right?
Exactly. Phloem transport, technically called translocation, moves the sugars produced during photosynthesis, the assimilates around the plant. It follows a source-to-sink pattern. A source is any part of the plant that's producing or releasing more sugar than it needs for itself. Usually, mature photosynthesizing leaves are the main sources. Storage organs releasing stored reserves, like a potato tuber sprouting, can also be sources. A sink is any part that needs sugars for growth or storage, think. Root tips, shoot tips, developing flowers, fruits, seeds, or storage organs that are filling up, like that potato tuber later in the season.
So the direction can change.
Absolutely. A leaf might be a source when it's mature, but it was a sink when it was young and growing. Roots might be sinks most of the time, but could become sources if they're exporting stored sugar. Developing fruits are very strong sinks, often drawing sugars from many different leaves.
How do we know sugars move specifically in the phloem sieve tubes?
Classic experiments like girdling or ringing a tree are key evidence. If you remove a ring of bark, which includes the phloem, down to the wood, the xylem, you see sugars accumulate above the ring, causing swelling. Things below the ring are starved.
Because you cut the downward pipeline.
Precisely. We can also use radioactive tracers. If you feed a leaf radioactive carbon dioxide, 14CO2, it gets incorporated into sugars during photosynthesis. You can then track where that radioactive sugar moves, and autoradiography shows it traveling within the phloem sieve tubes.
Clever. And what about aphids? You mentioned them helping with xylem research. Wait, was it xylem or phloem?
Phloem. Aphids are fantastic natural tools for studying phloem. They have these incredibly fine, needle-like mouth parts called stylets that they can insert directly into a single sieve tube cell.
Wow. Precision insects.
Totally. And the pressure inside the sieve tube is actually quite high, so the phloem sap is forced out through the stylet and into the aphid. Sometimes it comes out the other end as sugary honeydew.
So scientists collect the aphid's output.
Or even just the sap exuding from the stylet if the aphid is carefully detached. Analyzing this sap confirms it's mostly sucrose, often 10–25%, plus amino acids, hormones, some minerals. And the flow rates measured this way can be really fast, 50 to 100 centimeters per hour, much faster than diffusion.
Okay, so how does it move that fast? Not diffusion. What's the mechanism?
The widely accepted explanation is the pressure flow hypothesis, first proposed by Ernst Mach back in 1927. It's based entirely on osmosis creating a pressure gradient.
Osmosis again. Seems important in plants.
Fundamental. Here's how pressure flow works, step by step. 1. Phloem loading. At the source, say a leaf cell making sugar, sucrose is actively loaded into the phloem sieve tubes. This requires energy. Pumping sugar into the sieve tube makes the sap inside very concentrated.
Okay, high sugar concentration in the sieve tube at the source.
2. Water enters. This high sugar concentration lowers the water potential inside the sieve tube compared to the nearby xylem, so water moves by osmosis from the xylem into the sieve tube at the source end.
Inflating it, like the guard cells.
Exactly.
Dude.
This influx of water builds up high turgor pressure within the sieve tube at the source. 3. Bulk flow. Now you have high pressure at the source end and lower pressure at the sink end, where sugar is being removed. This pressure difference drives the entire solution of water and dissolved sugars through the sieve tubes from source to sink. It's a bulk flow like water through a pipe.
And at the sink.
4. Phloem unloading. At the sink, say a growing root tip, sugars are removed from the sieve tube either passively or actively to be used or stored. This makes the sap in the sieve tube less concentrated. 5. Water exits. With the sugar removed, the water potential inside the sieve tube at the sink becomes higher than in the surrounding tissues, often the xylem again. So water moves by osmosis out of the sieve tube at the sink.
And that water can go back into the xylem circulation as a whole cycle.
It is. Water circulates between xylem and phloem, facilitating the pressure gradient that drives sugar transport. Critically, the sieve tubes themselves are largely passive conduits for this long-distance flow, but the loading at the source, and often the unloading at the sink, require active transport and metabolic energy from the plant.
You mentioned loading requires energy. How does that sugar actually get from the leaf cell into the sieve tube?
There are a couple of main routes. In many herbaceous plants, it's apoplastic loading. Sucrose moves out into the cell wall space, the apoplast, near the phloem, and then it's actively pumped into the sieve tube companion cell complex. Companion cells are closely associated helper cells for sieve tubes.
So it takes a detour outside the cells first.
Yes, and that pumping step often involves a proton pump, using ATP energy, creating a proton gradient, and then a symporter protein that brings sucrose into the phloem, along with a proton flowing back down its gradient, like a revolving door powered by protons.
Yeah. What's the other?
Symplastic loading. Here, sugars move entirely through plasmodesmata, those cell-to-cell connections from the photosynthetic cells all the way into the sieve tubes. Some plants using this route have a neat trick called polymer trapping. In specialized intermediary cells next to the sieve tubes, sucrose is converted into larger sugars, like raffinose or stachios.
Why bigger sugars?
Because these larger sugars are too big to diffuse back out through the narrow plasmodesmata they came in through, but they can move forward into the larger sieve tube connections. It effectively traps the sugar near the phloem and maintains a concentration gradient driving diffusion towards the sieve tube. Still requires energy, but indirectly.
On some, just diffuse passively.
Yes, some trees seem to use passive symplastic loading. Sugars just move down their concentration gradient from the leaf cells into the phloem, where the flow itself maintains a lower concentration. No active pumping needed for loading itself.
So different strategies for loading. What about unloading at the sink? Is that active too?
It can be passive initially, just diffusing out down a concentration gradient. But often, the subsequent steps moving the sugar into the actual storage or growth cells of the sink tissue require energy. For example, to store huge amounts of sugar in a sugar beet root, the plant has to actively pump sucrose into the storage vacuoles, definitely requiring energy. In growing tissues, energy is needed just to maintain the gradient by quickly using up the arriving sugars.
Okay, let's try and wrap this up. We've gone from water loss being unavoidable for photosynthesis
Transpiration, yeah.
to the absolutely mind-bending cohesion tension theory, pulling water up hundreds of feet.
Powered by the sun and water's own stickiness.
Then into the roots, how they absorb water and actively grab nutrients, sometimes with fungal help.
The Casparian strip, checkpoint, root pressure, hydraulic redistribution, lots going on down there.
And finally, the phloem's pressure flow system, shipping sugars from source to sink using osmotic gradients.
Loading, flowing, unloading. An elegant system.
It really is. From tiny pores opening and closing to these massive flows throughout the whole plant. It definitely makes you look at plants, even common ones, with a bit more awe, doesn't it? Master engineers, truly.
Absolutely. And perhaps a final thought to leave you with. Consider this intricate network, largely powered by simple physics evaporation, cohesion, osmosis, and solar energy. How universal might these physical principles be for transport systems and life elsewhere? If life evolved under different conditions, different atmospheres, maybe without trees like ours, would similar physical solutions emerge? It makes you ponder the fundamental constraints and opportunities that physics places on biology.
That's a great question to ponder. Thank you for joining us on this deep dive into the hidden dynamic world inside plants. We hope you picked up some useful insights and enjoyed this shortcut to being well informed.